And so yesterday we talked about the direct loop and the indirect loop, remember? Okay, and we kind of functionally distinguish these two loops as the direct pathway as causing a disinhibition of this internal segment, the GP internal segment, which is tonically active and tonically suppressing the thalamus. So if you, now let's see if I get this right. Okay, so remember the internal segment is tonically active and tonically suppressing the thalamus. Okay, if the cortical inputs excite the striatum, that inhibits this tonically active internal segment, which disinhibits the thalamus, which triggers a motor plan or a movement of some kind, right? So that's the disinhibition that triggers movement. Okay, so direct pathway triggers movement. And then we learned that this indirect pathway, because we've got kind of got an extra negative step here, negative times negative is a positive. So if you activate the caudate putamen through this indirect pathway, you actually excite the internal segment, which just increases the inhibition, which suppresses the thalamus and suppresses movement. So direct activates movement, indirect is antagonist and suppresses movement. Okay, and I think, then we have this extra pathway here, which also reinforces the excitation of the internal segment and further suppresses movement. So you've got sort of multiple ways of suppressing movement. And I think anybody who looks at this says, oh, why is it so complicated, right? We've got these multiple loops, we've got negatives on negatives, and you wonder why is it like this? And I wanted to kind of come up with an analogy for why you would even design something like this. So how many people do programming? Oh my gosh, you have to learn how to do it. Okay, who knows what programming is? Okay, so basically you're giving the computer a set of instructions, right? And let me say, if you go into graduate school, you're going to have to learn how to program one way or another, so be warned. Okay, so you're giving a set of instructions. So you have some sort of input, okay, and you're using it to instruct some sort of output. Right? And so this is basically what we have. Say the cortex is input and the activation of the motor cortex is your output. Okay, but say you have a couple of options. You have output A, and maybe you have output B. Okay, and so you have to have some way to choose between those outputs. So you put a little box, right? So you say, if something, then go this way, otherwise go that way. Okay, so you put in a little box to do maybe a comparison. So maybe you have two types of input, okay? And you want to compare them, and that tells you to do output A if you've got more of this, and output B if you've got more of this. But the important thing is you have to stick a box in here, right? Okay. So let's say, you have to come up with some sort of motor plan, and you have a couple of inputs that you're comparing. But this if statement is going to depend on, say, context. Say you want to do this one thing in the day and something else in the night. Okay, so now you have to have another piece of information that affects this box. Okay, so now you've added another component. Okay, and say you want to adjust what you're doing kind of as you go along, and now you need some feedback. Okay, so now you need output B to feed back in, and output A to feed back in. Okay, is this starting to look familiar? Okay, so this is basically what's going on here, is you have multiple kinds of inputs that you're performing some sort of computation on. It has to be context dependent, so you have to kind of, pull in information from other parts of the brain, say. And you need to add in some feedback. So it's very easy to see that this is the kind of structure you will start to end up with. And the sign on each of these just depends on how you're going to kind of combine these signals, right? Does that make more sense to everybody? Okay, so that's one thing. So that's kind of why you have these sort of complicated circuits. The second point is, we talked about, well, why do you have a direct line that's ultimately excitatory and an indirect line that's ultimately suppressive? You're trying to do these two different things, but, you end up canceling out. What's the point? And I think to answer that question, you have to consider this sort of multiple output concept. That you have these circuits, and let's take this flat sheet, let's say there's a flat sheet of circuits, but for every output, you have to have a duplicate circuit. Okay? And then you have interactions between those circuits. So if you have action B, you actually want to suppress action A, right? You're trying to choose among a very wide range of options. And so that's where that comes in. We're sort of neglecting that component here. Interactions between multiple versions of these direct and indirect pathways. So anyway, so I just wanted to give you guys a little bit of an intuition about why we're even studying these kind of complicated circuits. And I think the third thing I wanted to say was all the explanations I'm giving you are really hand-wavy, aren't they? Right? So I'm saying, oh, there's a motor plan, oh, there's some sensory input. And we're not really defining exactly what those things are we? And the reason is that the farther you get from direct sensory inputs and the farther you get from direct motor outputs, things get fuzzy, right? So this is a sensory motor circuit. It's neither just sensory or just motor. And because we can't really define the inputs and outputs strictly in terms of sensory or strictly in terms of motor, it gets a little fuzzier. And this is one reason that people like me study sensory coding, because this is sort of too fuzzy for my personal taste, let's say. All right, so, and then the last thing I wanted to remind you was we're going to start talking about these dopaminergic inputs. So we said yesterday that there's a dopaminergic input from the substantia nigra pars compacta, but it has a disparate effect depending on the receptors that are expressed in the neurons in the direct versus indirect pathway. So the direct pathway neurons express a D1 receptor, and dopamine has an excitatory effect. The indirect neurons in the striatum have D2 receptors and are suppressed by dopamine. So you apply dopamine, you bias the circuits in the direction of enhanced direct activation. And I suppose the opposite, if you tip dopamine away, you would bias the circuits in the other direction. Okay. And so this leads us to two of the sort of best known neurodegenerative diseases. Let's see. Oh, here's some anatomy of this dopaminergic input. So here, remember here we talked about this is the striatum, the caudate putamen. Here's the globus pallidus. Here's thalamus. And down here in the midbrain is the substantia nigra. And there's one component, the pars compacta, that supplies this dopaminergic input, this green line right here. Okay, this is seen if you do a cut through the midbrain as sort of this dark banded area. So substantia nigra, or dark black, and so on. And when it's and in Parkinson's, you have the loss of these neurons. So you see this dark banding has been diminished. So in a way, this is a very particular effect on the system. Okay, and this has a result of causing Parkinson's disease, which everybody's heard of. It was early described in the early 19th century by a guy named James Parkinson. Okay, and described as a shaking palsy. It has very particular clinical signs that I think everyone's somewhat familiar with. Tremor, rigidity, slowed movement, known as bradykinesia, freezing, loss of postural reflexes, flexed posture. And these all together are termed hypokinetic. So there's sort of a loss of movement, kind of a locking in of movement. Okay. And we said the pathology of this, the neuropathology, is the loss of those dopaminergic neurons in the substantia nigra. Okay. So what effect, how does the circuit explain that effect? And so the basic idea is that these dopaminergic neurons are, said has an enhanced effect, are diminished. This component of the circuit is degenerated. Okay? And the loss of this dopamine has the effect of tipping the balance toward more inhibition coming out of the internal segment. So generally the end effect is that you have more tonic inhibition. So this affects both the direct and the indirect, but something about the way the system is set up means that it diminishes the direct pathway and increases the indirect pathway, which then enhances the internal segment activity, which means you have more inhibition, which basically puts a brake on movement. Okay, so very common treatment for Parkinson's disease. I mean, ultimately, it's a neurodegenerative disease that kills people, but there's at least some degree of treatment by using L-DOPA. So remember, this is the dopamine pathway. Here's the dopamine. Here's DOPA. So if you supply the precursor for the dopamine, you can enhance levels of dopamine. Or you can use monoamine oxidase inhibitor. So monoamine oxidase breaks down dopamine. So if you prevent the breakdown of dopamine, you can recover from Parkinsonian symptoms. Okay, and then other things people are trying are dopamine receptor agonists. Another attack on this sort of neurodegenerative disease is to use deep brain stimulation. Okay, how many people have heard of deep brain stimulation? Yeah, sort of, kind of. So it's, it was initially a little controversial, but I think has become sort of more of an accepted treatment. It seems very invasive, but I'll show you in fact, right now we'll show you some of the effect of this. So let's take a look at that. Okay, so first we're going to look at a video of a patient that's being treated with L-DOPA.
With shuffling and elements of fascination.
It is not a true no.
Hang on. Too many videos.
In this first clip, we're examining the gait of a 55-year-old man with Parkinson's disease during the off phase. Notice his typical Parkinsonian gait with shuffling and elements of fascination. In addition, notice his reduced arm swing.
So he's got his arms sort of rigidly by the sides of his body, whereas normally you would move your arms.
Notice how difficult he finds to turn when asked to do so by the examiner.
So he's trying really, really hard to move, and it's hard.
In the second clip, we're looking at the same patient about 30 minutes later after he's taken his L-DOPA medication. There is a dramatic improvement in his gait, but if you look closely, it also demonstrates some mild dyskinesias of his head and neck as his head wobbles a little bit from side to side.
Right, so that's pretty remarkable. That's not, the next day or a week later. That's minutes later. Which is crazy. Right, so here's a video of a gentleman with severe Parkinsonian tremor, and he's going to describe what it's like to have deep brain stimulation. So he's got the electrode already implanted, and he's got a thing in his hand that he can control the stimulator himself.
is placed into the brain, I understand, comes level with the top of the nose and the ear. And it is connected to a battery in my chest, in the skin of my chest here. And this is controlled. I can control the amount of the voltage going in, I can control the length of time that the pulse is, and I can identify the number of times per second that it goes in. And I'll turn myself off now. Because my concentration is up to 90% of my tremor, I'm trying to control it. And I'm thinking of the word Parkinson's all the time. I'm not terribly, I'm not concentrating on my conversation with you at all. It's quite.
All right. So quite effective. Right, and you can see why if you had that level of tremor, why you might be interested in doing something as extreme as implanting an electrode in the middle of the brain. Yes.
If you're losing these dopaminergic tips and you're increasing tonic inhibition of motor output, then how come in his case he had such violent tremors?
Yeah. So that seems like it's activating movement. And I think here, this is where the sort of balance between the two come. Do you remember the robot movie? Where there was a point where the robot was basically shaking. And I think that has to do with improper activation of movement, or you're activating two competing
can you explain which robot movie?
Oh, this was the one with the robots, the silly one with all the robots falling down. There was one where the robot was basically trying to get out of the car and just started shaking.
Oh, that one.
Yeah. The other ones they would just fell down. So not the scary dog robot, but the sort of more human robot. And I think that it has to do with the symptoms are going to vary from person to person. But just like you need to have proper opponency of your muscles, right, to flex versus extend, you need to have the proper opponency of these different motor plans. If you sort of activate all of them at once, or try to suppress all of them at once, you end up with this sort of shaking thing. It's not a very good explanation, but it's kind of the best I can do. And one question is, why does deep brain stimulation work at all? Right, so the dopamine thing you can kind of understand if you have sort of flood the brain with dopamine, you sort of it gets back to its sort of baseline levels. With the deep brain stimulation, the stimulator is often in either the internal segment of the lobus pallidus or in the subthalamic nucleus. But why does stimulating it work? And I think the basic idea is that you're kind of resetting the circuit, just like a pacemaker in a heart kind of resets the sort of normal rhythmic activity in the heart muscles, that this basically resets the normal activity patterns in the lobus pallidus. Okay, so that is Parkinson's. So at least, with Parkinson's you have some degree of treatment. You can treat the symptoms, if not the degeneration itself. Not yet, anyway. Huntington's disease is a little bit different. It's a much more sort of broad-based degeneration. It was originally described by George Huntington, so all these diseases named after a person who described them. It has clinical signs that are sort of the opposite of Parkinson's. And so the idea is that mostly you have hyperkinetic motor symptoms. And I'm sorry, I don't have a video of this, but you'll see that with Huntington's people, that oftentimes they'll have to like swing their arms or make movements that they just can't control. So they have these rapid, jerky motions called choreiform movements. And then you also have mood and cognitive disorders, which probably has a lot to do with the cortical degeneration that I'm about to show you. So there's a very profound degeneration of the striatum, the caudate and putamen, but also of the frontal and temporal cortices. And it's a genetic disorder, right? So it's a, let's see, what is it? So an autosomal dominant. So if you have one of the defective genes, you are very, very, very likely to get Huntington's. And this is genetic testing, and so it leads to kind of a conundrum for people. If you have Huntington's in your family, do you get tested to see whether or not you have the gene? Because once you have the gene, you will get Huntington's. And lots of people say, I just don't want to know. Okay, so you can think for yourself whether this is something that you would want to know. If you have a 50-50 chance of inheriting from an affected parent, is that something you would want to know? And the worst part is that there's just no treatment. Unlike Parkinson's, there's just not much that you can do. You can treat the symptoms to some degree. You can use sort of the usual battery of antidepressants or antipsychotics to deal with the cognitive dimension of the dementia. But this is what happens to a brain of a Huntington's patient. So here's sort of normal striatum, and this is the greatly diminished striatum. But you'll notice that there's also just an enormous loss of cortical real estate here. So that's sort of a particularly devastating part. Okay, so the effect on the basal ganglia circuit is that there's selective loss of the indirect pathway. Okay, so you still have a direct pathway, okay, but the indirect pathway is degenerated here. This inhibition from the striatum to the external segment. And that means that the external segment is excited, sort of in a more excited state, which then has a suppressive effect on the internal segment. Okay, so in the end, there's less tonic inhibition, and this allows really inappropriate gating of movement, and that's why you get those sorts of jerky muscles, because you can't suppress those inappropriate motor plans. Okay, so loss of striatal input to the external segment tips the balance toward less inhibition of the internal segment, and that's when you get this inappropriate motor activity. Okay, so that's Parkinson's and Huntington's, and so we've talked almost exclusively about motor loops of the basal ganglia, and there are two other aspects that we're really not going to talk about, but you should probably be aware that there are additional loops that deal with the prefrontal cortex. So you have the prefrontal cortex, so we're not going to talk a lot about this, but the prefrontal cortex, ahead of the sort of premotor regions, are really involved in what we would call executive function, decision making, cognitive processing, working memory, planning, attention. An example of what's thought to be a deficit of this prefrontal basal ganglion loop is Tourette syndrome. So you have vocal and motor tics, and the idea is that it involves some speech regions in the frontal cortex. And then a second non-motor loop is known as the limbic loop. So this is involved in emotion, motivation, reward, addiction. Involves a slightly different portion of the striatum. And an example of what's thought to be dysfunction of this loop is schizophrenia. Okay, so this has to do with an affective disorder. And one of the ways to deal with this is to use antipsychotic drugs that act on dopamine receptors. So maybe there's something wrong with the dopaminergic part of the circuit that causes schizophrenia. Okay. And now you can sort of relax your brain a little bit. Because the anatomy of this area is kind of hard to picture and see, there's a nice video summary of the basal ganglia that I wanted to play for you. So let's see. All right, so you don't have to take notes. This is just to give you good visualization.
It is intuitive to understand how sensory information gets to the brain and how motor information can travel to muscles. Together, these two systems allow us to detect and respond to the world around us. But how do we engage with that world? How do we determine what is important? How do we fall in love? These higher cortical functions involve the complex interplay between neurotransmitters and hormones throughout the entire nervous system. There are two major anatomical substrates that influence these behaviors, the limbic system and the hypothalamus. Today, we're going to take a closer look at these structures.
Hang on. That's not the right one. I thought that was weird when she started talking so long.
Deep in the forebrain sit a group of nuclei that integrate all cortical activity into one behavioral output. These are the basal ganglia. These deep nuclei are interconnected with each other and with all areas of the CNS. A series of parallel circuits regulates different aspects of behavior. One circuit is primarily involved in the integration of motor output. Another circuit plays a major role in learning and cognition. And finally, a third circuit regulates emotional aspects of behavior. So what does this mean? You can tell when someone's having a good day or a bad day by their posture, body language, and even their tone of voice. The sum of all experiences, hours of practice, memory, emotion, reward seeking, and the plan for a particular movement are all integrated and result in one behavioral output due to the coordinated activity within the basal ganglia. Now let's go and have a look how these structures work together to influence our behavior. The goal of all movements is that they be fluid, efficient, and target-oriented. In order to achieve this, motor, association, and sensory cortices send their input to the basal ganglia, essentially asking them to make a decision of whether or not to execute this movement, and then streamline the plan to do so. While one pathway will facilitate goal-oriented movements, another one will suppress all competing movement. This results in motor output that is sleek and appears effortless. This motor loop is active primarily when a task has been well learned. Together with association areas of the cortex, it helps to put together a routine for established motor output. This is the circuit that is active in these dancers now as they perform a well-rehearsed, fluid routine. Afferents from motor and sensory areas of the cortex reach the putamen. Here, the information is processed. All movements that are part of the integrated plan are facilitated through the direct pathway, and all competing movements are inhibited through the indirect pathway. The signal that reaches the thalamus is a balance of these pathways.
Okay, what's she omitting here?
What?
Which thing is she missing between putamen and thalamus? Lobus pallidus.
From here, projections back to the motor cortex result in measured and coordinated output. The cognitive or association loop is particularly important for higher cortical functions and motor learning. We try out different strategies for accomplishing a desired movement and think about the different possibilities. Once we have refined our movements, the activity in this loop will decrease and the motor loop will take over. The cognitive loop is active in the hours of training and rehearsing these dancers have gone through to achieve a beautiful performance. The cognitive loop is this training loop. Afferents from various cortical areas reach the caudate nucleus, and in particular the nucleus accumbens, which contains reward-affirming dopaminergic connections. These connections provide the rewarding feeling following the successful completion of a task. From the nucleus accumbens and caudate, projections reach the thalamus via the direct and indirect pathways. The thalamus, in turn, projects back to the cortical areas. You can almost consider this circuit to be a consulting service that streamlines cognitive processes by separating the successful ones from the unsuccessful ones during learning. Finally, the limbic circuit links the basal ganglia with areas of the brain that process emotionality. Postures, gestures, and facial expressions related to emotion are mediated by this circuit. Everyone around us can pick up on these emotions that we display as we go through our day. The motor expression of emotions is evident in everything we do. In the limbic circuit, the hippocampus, the amygdala, and the limbic lobe all project to the caudate nucleus, and again, the nucleus accumbens with its dopaminergic projections. From here, the direct and indirect pathways project to the thalamus as to which behaviors to facilitate and which ones to inhibit. The thalamus, of course, projects back to the limbic areas of the cortex. This circuit adds the emotional component to behavior and colors all of our cortical output, from motor performance to cognition. Here we have dissected away parts of the lateral surface of the cortex to show the corona radiata as it projects to all area of the brain. This here is the insula. Underneath the insula lie the putamen and the globus pallidus. The putamen, together with the caudate nucleus, is called the striatum and is the main input to the basal ganglia. You can see the fibers of the corona radiata converging behind these nuclei to form the internal capsule. Here we have removed the putamen and globus pallidus to show the internal capsule. You can see the anterior limb and the posterior limb of the internal capsule. The caudate nucleus lies deep to the internal capsule. It is best shown on this medial view of the brain. Here we have opened up the lateral ventricle from a medial aspect. This bulge here, in the floor of the anterior horn of the lateral ventricle, is the caudate nucleus. Its body follows the curvature of the ventricles into the roof of the inferior horn. Here we have removed the lining of the ventricles to show you the caudate nucleus as it swings around, following the shape of the lateral ventricle. There are two additional structures that play an important role in the basal ganglia circuitry: the substantia nigra and the subthalamic nucleus. The substantia nigra is located in the cerebral peduncles of the midbrain and can be easily found due to its black pigmentation of the dopaminergic neurons. These neurons project to the putamen and caudate to influence motor output. Additionally, dopamine release from the substantia nigra facilitates cortical outputs and feelings of reward. The subthalamic nucleus, which you can see right here, functions like a pacemaker for the basal ganglia circuitry, defining the rhythm of output through inhibitory influences. It is located inferiorly and a bit lateral to the thalamus. So you can see in this coronal section. So here's the thalamus, and this is the subthalamic nucleus. Let's have a look at the anatomy of all nuclei in cross-section. This is how we commonly view these structures using MRI or CT scanning technologies. This is a coronal section through the brain. Let's have a look at our landmarks. Here's corpus callosum. These are the lateral ventricles. This here is the third ventricle, and the thalamus is on either side of the third ventricle. And here's the inferior horn of the lateral ventricle. This bundle of projection fibers is the internal capsule. And here is on the other side. Here, this is the internal capsule. It's the posterior limb of the internal capsule because it separates the thalamus from the putamen and globus pallidus. Here in the floor of the lateral ventricle is the body of the caudate nucleus. Here, lateral to the internal capsule, is the putamen and globus pallidus with its internal and external parts. Now let's have a look at these nuclei in a horizontal section. Let's begin with our landmarks. Again, this here is corpus callosum, that big commissural fiber bundle, and this is the posterior aspect of corpus callosum, also called forceps major. These here are the lateral ventricles. Here's the anterior horn of the lateral ventricle on either side, and here's the posterior horn. This nucleus here is the thalamus, and here's the thalamus on the other side. Now the thalamus is separated from the putamen and globus pallidus through the posterior limb of the internal capsule. And here's the posterior limb on the other side. The anterior limb of the internal capsule separates the head of the caudate nucleus from the putamen and globus pallidus. So here's the anterior limb of the internal capsule, and here's the anterior limb of the internal capsule on the other side. This here, in the floor of the anterior horn of the lateral ventricle, is the head of the caudate nucleus. Here's the head of the caudate nucleus on the other side. This here is the globus pallidus with its internal and external parts. And this here is the putamen. Lateral to the putamen is the cortex of the insula. Now what's really interesting on this section is you can see that the head of the caudate and the putamen are fused here. And this is the nucleus accumbens. A solid understanding of the alignment of these nuclei in both the coronal and horizontal planes is a key skill in interpreting cross-sectional anatomy in all types of neuroimaging. All of our cortical output is integrated in the basal ganglia. None of these loops works independently. Everything needs to feel right in order to have the perfect performance. The basal ganglia are like a funnel that takes in all of our cortical activity. They integrate this activity with our motivation, our goals, and our general mood, and result in one behavioral output.
Alright, that was very soothing, wasn't it?
I'm tired now.
I'm not so sure about the funnel analogy, but it's always cool to see somebody who will cut a human brain like a slice of bread. It's always fun. Yes?
So one thing she talked about was the selectivity of the basal ganglia through the direct and indirect pathway, like the type of motor output that they choose or not choose. So would an example of that be like, say you had like an animal, right, and it wants to mate or something, would it promote mating behavioral outputs through the direct pathway and suppress maybe outputs that correspond with scavenging for food or maybe sleeping in the indirect pathway? Is that sort of what she was getting at when she talked about the funny?
I think that's kind of a higher level order of behavior. So the question was, is the kind of behavioral choice that you're making, like whether you're going to mate or go find food, you know, or mate or fight, right? And I think that might be sort of a higher order decision that unfolds over a slower period of time. I think that more she's talking about sort of immediate movement.
Yeah, I'm saying like after you've made the decision to mate, like when you are engaging in mating behavior or engaging in a particular behavior, the direct pathway would then promote behaviors that are pertinent to whatever you're engaging in, and then suppress other output that maybe don't correspond with that. Is that sort of how it's working?
I think that example is maybe too high level.
Okay.
Organization of behavior. That's sort of, and I think that what we're talking about is more of a sort of next level. Like, are you going to approach the content-specific?
Okay, so then
or are you going to make a vocal call in this direction or in that direction? I think those sort of more like local decisions. It's still complex behavior and movement, but it's not maybe not necessarily the advanced planning.
Okay, so you're saying like it's just approaching someone.
Yeah,
that
because I think what you're talking about is more of an executive function.
If I'm approaching someone, that would be direct pathway, and any other motor movements that don't actually relate to me approaching or specific behavioral action would be coded in the indirect suppression of that.
Yeah, I think so.
Okay.
Yeah. There's another question back. Yes.
Sorry, I was just fixing.
Oh, okay, you were
sorry.
You just had a motor plan. Yeah. Okay, so let's move on. So that's it for basal ganglia.
Yeah.
And I think you can see that there's a lot left to know. Okay, so we're going to move on to the sort of final piece of the overall motor puzzle, and that's the cerebellum. Okay, so remember this whole upper motor neurons of motor cortex and brainstem. So we talked about how basal ganglia influences the activity here. Now we're going to talk a little bit about cerebellum. Okay. Cerebellum can have influences both on brainstem centers and also on motor cortex. Okay, and the sort of distinction between what basal ganglia and what cerebellum is, the current thinking. So we talked about basal ganglia as making decisions about which motor movement, which motor plan are you going to execute, and suppressing all the others. And in the video, she talked a little bit about learning, but I'm going to present the idea here that cerebellum is an important component to motor learning because it has, the theory now is that it has a function in error correction. So it's really important for motor skill learning because you have to try something and then correct for your errors, and in that way learn complicated movements. And so there's a distinction between those But obviously, everything has to kind of work together. Okay, so we all know cerebellum as the major component of the human brain. Varies in sizes in different animals. Birds in particular, really big cerebellum. But in humans, this is sort of a ventral view of the human brain. So here's the cerebellum. And in humans, it has a relatively small volume compared to the cortex, the forebrain, but it has at least as many neurons. So it's very densely packed with neurons. Okay. Here's a sagittal section. So this was a ventral view. This is the brainstem right here. And so you see the two hemispheres of the cerebellum. This is a sagittal view. So again, you sort of cut the brain directly in half, and you're looking from the medial side. Here's the corpus callosum. Here's the brainstem and spinal cord. This is the pons, this sort of bulby component here. And here's the cerebellum. And what you don't really see here is the peduncles. So there are these two dense superhighways that go up to from about this level, kind of midbrainish, to the cerebellum that you don't really see. But what you can see really well is this cut through the cerebellar hemisphere. You can see that you have this sort of folding. So just like the cerebral cortex, you have these very dense folds in the layered structure of the cerebellum. Okay, and these are known as folia in cerebellum. You can live without your cerebellum, so this led people to think for a long time that you didn't really need it, which didn't really make any sense, right? You don't waste all of those neurons on nothing. But it doesn't kill you. What you do have is if you're missing your cerebellum, this is shown in this MRI, so this blank spot right here is where the cerebellum should be. If you're missing the cerebellum, what you have are motor problems. Late walking, ataxia, and so on. But you can make movements, but there are definite problems with coordination and learning. Okay, so here's another view of the cerebellum. This is now looking from on top. So say you've removed part of the cerebrum. Looks a little like a lump of hamburger here. Not very appetizing. But here's a cartoon. So what you can see is that there's a sort of central area here, this sort of central little ridge that's called the vermis. Okay, vermis for worm. So it's a little like a worm, sort of. And then on either side you have the two hemispheres of the cerebellum. So again, this is sort of posterior is down, anterior is up. So if you take this sort of dorsal view, remember this is sort of folded around, it's like a little ball, sort of folded around. So if you sort of take that and unfold it and flatten it out, you get something that looks a little like this. So here's the top edge and the bottom edge. Again, you have the vermis, the ridge in the middle, and the two hemispheres, and now you can see a little appendage down here that's known as the vestibulocerebellum. So there's sort of three main components here. You have what's called the cerebrocerebellum, so sort of cerebral-like cerebellum, which is on the lateral parts of the hemispheres. You have what's termed the spinocerebellum, which includes the vermis, but also a little bit of the hemispheres, so sort of this middle strip here. And then this little appendage down here is the vestibulocerebellum. Okay. And functionally, we'll start with the vestibulocerebellum, kind of obviously. This is involved in vestibular motor function. Very descriptive. And then the cerebrocerebellum and the spinocerebellum kind of have a similar organization. Remember we talked about spinal cord having sort of medial and lateral organization? Well, you see this again in the cerebellum. So the medial component, which is the spinocerebellum, especially along the midline ridge, is involved in movement control of proximal muscles. So sort of postural midline stuff. And as you move lateral into the cerebrocerebellum, you get more and more distal control and fine motor control of your hands. Okay, so here is another view of the cerebellum. So again, we're looking kind of dorsally. Here's the surface of the cerebellum. And you probably, what's lying underneath it should be very familiar because we've often seen this component of the midbrain here, right? There's the thalamus, and here's the striatum. So this is sort of central forebrain, midbrain, superior colliculus. And we've seen this a lot in terms of basically where we have cut away the cerebellum. So a bunch of past slides have looked. But now we've stuck the cerebellum back on there. Here's the cerebellar peduncle. Okay, and this is just where you have inputs and outputs, obviously. And then, importantly, there are these deep cerebellar nuclei. So not only do you have those folds within the structure of the cerebellum, you have these very important nuclei that serve as the main output center. Okay, so projections to the cerebellum. Again, it receives a pretty complicated set of inputs that we'll try to summarize kind of simply. So first, let's talk about the pontine nuclei. So remember, those are the pons, the nuclei and the pons. And those provide an indirect input from cerebral cortex. So there's a relay from cerebral cortex. But importantly, this is a contralateral decussation. Okay, so the pontine nuclei cross over and project to the opposite hemisphere of the cerebellum. So remember, cerebral cortex receives inputs from the opposite side of the body and controls outputs to the opposite side of the body. So now, if this cortical input crosses over to the cerebellum on the opposite side. Okay, so now the cerebellum is receiving input that deals with the same side of the body. Did everybody follow that? In contrast, the vestibular and proprioceptive systems bring in sensory input from the ipsilateral side. Okay, so brainstem inputs are ipsilaterally projecting to the cerebellum. Okay, but importantly, these are all from that side of the body as well, at the level of the brainstem. Okay, and then finally, there's an input from the inferior olive. We'll talk about in a few minutes. And then the projections out are these deep cerebellar nuclei. Yes?
I was wondering if you could go over that contralateral decussation.
It'll come up in the next couple of slides. Yeah, thanks. Okay, so here's the same thing kind of anatomically. Sources of input are these pontine nuclei, vestibular nuclei. Here's the inferior olive. These are these proprioceptive inputs. Remember the dorsal nucleus of Clark, sort of, kind of. This is one of the dorsal column nuclei, the cuneate nucleus. Okay, we talked about that in terms of proprioception. So you have proprioception, vestibular, cortical inputs projecting into the cerebellum. Okay, so remember proprioceptive. All right, so this is sort of another map that sort of explicitly maps the inputs. Okay, so here we have cortical inputs going through the pontine, and here's this decussation crossing over, right? Okay, and inferior olive also sending inputs, and then here are the brainstem nuclei that are also sending inputs. Okay, so here's cortex, goes to the pontine goes to cerebellum, and coming up. Okay, so the cerebellum really deals with information on the same side of the body. Okay, and so this is different from cerebral cortex that generally deals with information on the opposite side. Okay, so when it sends outputs, generally if it's dealing with motor outputs, it's going to send them back out to the same side of the body. Okay. So cerebellum, is it sensory, is it motor? It's kind of both. It's certainly true that if you record from cerebellum and you do different types of sensory stimulation, that you get responses. Typically, we talk about the cerebellum as having somatosensory maps. So like the somatosensory cortex, you have maps of body space that maps directly onto the layers of the cerebellum. But it's a fractured map. You actually have multiple maps sort of in different spots. It not only gets somatosensory input, it gets some other input. We talked about vestibular and proprioceptive. I can give you firsthand evidence that you can also get auditory inputs, so it really kind of gets a mishmash of different inputs. Okay. And the outputs, we talked about each of these components of the cerebellum having different outputs. So the vestibulocerebellum projects back to vestibular nuclei, which then also sends motor outputs. These two components go through the deep cerebellar nuclei and ultimately through thalamus to project back to cortex. So it kind of sends some outputs in one direction and other outputs in the other direction. Okay, the principal neurons of the cerebellum are these sort of really famous Purkinje cells. And we talked about these very, very early on as being these neurons that have this very elaborate branching pattern, right? So it has these very, very elaborate dendritic patterns. But interestingly, they're very flat. So they're like a little fan. So you've got these dendritic patterns, but it's very narrow. And so you have a whole series of these Purkinje cells that have their fan dendrites kind of all lined up, kind of all stacked up in the folia, in those folds of the cerebellum. Okay. And this is a really kind of a very famous circuit. We're still trying to figure out what exactly this circuit is doing. So it has a sort of this canonical circuit. So no matter where you go in the cerebellum, you're going to see sort of the same circuit over and over again. So the idea is that there's some computation that's going on, that it's doing something similar to all the information that it's getting, but what exactly that is still something that's being worked out. Okay, but we know a lot about the particular, cellular circuitry in this system. Okay, so we have Purkinje cells, and one interesting thing about these Purkinje cells is that they're inhibitory. So this is an example of sort of the major principal neuron in a system being an inhibitory neuron rather than excitatory. It receives multiple types of input. They have very large cell bodies that comprise the Purkinje cell layer. And then the other major cell are granule cells. So they're much smaller, but they're much more numerous. Lots and lots and lots of them. Okay, these are excitatory, and they send input to the Purkinjes via what we call the parallel fibers. And we'll take a look at the diagram of this in the next slide. And their cell bodies form the granule layer. Okay, so there's basically kind of a three-layered structure, as we'll see. Yeah, let's see the next slide. Okay, so here's kind of a canonical view of the cerebellum. So here's the surface of the cerebellum, and we've kind of cut away in two directions to make this little block. So there are three layers. There's the molecular layer, which is most superficial, the Purkinje cell layer, which is where this Purkinje cell bodies lie, and then there's the granule cell layer, which is where the granule cell bodies lie. And the molecular layer is interesting because that's where all these dendrites are. Okay, and the granule cells. Here we are. Here's a little granule cell. Sends an axon superficially into the molecular layer, and then it bifurcates, and then sends an axon in either direction that courses perpendicularly, or well, I guess parallel to the surface of the brain, perpendicular to the fans of the Purkinje cells, and kind of like a little, telephone wire system, just sends axons far and wide. And as a result, these contact lots and lots and lots of different Purkinje cells. Okay? And at the same time, Purkinje cells basically receive lots and lots and lots of parallel fibers. So this whole sort of neuropil, this layer neuropil, is just sort of filled with these coursing parallel fibers. Okay, so those Purkinje cells are receiving lots and lots and lots of input from a big population of these granule-cell outputs. Okay. Now the granule cells themselves are getting inputs from the mossy fibers. And then there's a second excitatory, and these are excitatory. There's a second excitatory input from what we call the climbing fibers. Okay, and these are derived from the inferior olive. And I have a text slide next that kind of outlines all this, so don't worry if you're not getting this all down. Just kind of look at the diagram. We have lots of parallel fibers, but each Purkinje cell is receiving input from a small number of what's called climbing fibers that wrap around these dendrites and have this incredibly strong excitatory input. Okay? And so this is kind of a vertical dimension. So you have kind of a horizontal dimension, and then you have a vertical dimension. Okay, so kind of compare and contrast these two types of input. Okay. On the one hand, you have the mossy fibers. The source of the mossy fibers are the pontine nuclei, which receives input from the cortex, spinal cord or vestibular system. These are indirect excitatory. Okay, the mossy fibers contact the granule cells that then form those parallel fibers to the Purkinje cells, and they run parallel to the surface of the brain and parallel and perpendicular to the fan of the Purkinje cells. Okay, so mossy fibers to granule cell parallel fibers to Purkinje. The climbing fibers, the source is the inferior olive, relaying cortical info. It's a direct excitatory. They actually literally sort of climb around those Purkinje cell dendrites and run in a vertical dimension and make this very powerful contact. Both of these actually also make excitatory inputs to the deep cerebellum nuclei. Okay. So, just to kind of give you an intuition, there's no deep consensus on precisely what this computation is. But let me give you sort of a little idea that you can think of this as anytime you have a system where you have a very distributed series of inputs where inputs contact multiple targets in very small ways. This is what we would sort of think of as an association cortex, right? So each one of these neurons is basically receiving lots of input and is kind of integrating over a large number of inputs. And each of these fibers is probably contacting relatively sparsely a large number of neurons. So in that sense, kind of think back to the piriform cortex that we talked about as being an association cortex. You have this sort of sparse distributed system. In contrast, this climbing fiber system is very direct, right? So that is, I'm contacting this one Purkinje cell. There's sort of a one-to-one strong connection. And so you could come up with some idea that you're trying to make some sort of comparison between sort of a very specific input and a general survey. Okay? And in this way, you can come up with this concept that you're trying to make some sort of a comparison. And the general idea is that you're trying to correct for some error, making a comparison and then adjusting based on that comparison. Okay. So what's the evidence that the cerebellum is involved in motor learning and plasticity? And we're going to look at a specific example. And hang with me. We're going to get through this in 5 minutes. Okay? But you got to follow, kind of stay focused. Okay. So the idea is that we're going to use an experience-dependent plasticity of saccadic eye movement. Okay. Now you all remember from the vestibular ocular reflex how you control your eyes, right? you move your eyes, you have to use the rectus muscles. So you have the lateral rectus and the medial rectus. Okay, so we're going to take advantage of this system. And remember that we talked about how these eye movements are coordinated, right? So they're kind of cross-linked so that you move them together. Okay, so we're going to take one eyeball and we're going to kind of nick the rectus just a little bit, just to make it a little bit weaker, okay? And we're going to leave the right eye just normal. Okay, so normally when you move your eyes, they move together. Okay, so here we're going to plot the position of the left eye, okay, versus time, and this is the position of the right eye versus time. So normally, if you're going to move your eye, you're going to move the position, get to the position you want, and stay there. Okay, if you weaken this eye, it just doesn't quite get to the target. Okay, so whatever the signal is that causes the saccade, you're going to move a little bit, but you don't quite get there. And now, if you do that, if you cover that eye with a patch, okay, and so now you're using the visual stimulus, and your right eye can see it, so your right eye moves over, and it's trying to move the left eye. So it's sending a signal to both. The right eye is successfully saccading to the appropriate position, but the left eye isn't quite making it. But you don't know that left eye isn't making it because you don't have any sensory feedback in that eye. Okay. So when you have a patch on the left eye, it's always undershooting. Okay. The position of the left eye is always undershooting. Now, if you move the patch back to the right eye, and you're trying to do the same task, your left eye doesn't quite move, and you know that you've made a mistake. Okay, you're not reaching the target. And so here, you're trying to sort of make little saccades to kind of correct and get to the right target. Now, your brainstem is sending signals to both of those eye control muscles. And so as it's trying to correct the left, it's over-correcting on the right. So the right is nice and strong. So it ups the signal in order to correct for the left, and then the right overshoots. Okay. And so over time, okay, 5 days after you've been patching and you've been making these corrections, your cerebellum has been rewired so that now instead of having to make these little small corrective motions, it just goes directly to the right spot. So it's like cranked up the signal to send your left eye to the right spot, the correct spot. And you patched over the right eye, so you don't know that your right eye is overshooting. Okay, and so after time, this gain on the signal has gone up, but it's gone up on both sides, so you see this overcorrection, this overshooting in the right eye. Okay, so that's when you have your cerebellum. That's what's going on. And the evidence that you need your cerebellum is if you take away your cerebellum or you damage the cerebellum, that you can prevent this type of motor learning. Okay. So 2 other things. We have 1 last minute. Chronic alcohol abuse causes damage to your cerebellum. Okay, so don't drink too much. But this is kind of what causes some of the ataxia associated with alcoholism. And there are a couple of genetic mutations that cause disorganization in the development of the cerebellum. Wheeler and Weaver causes developmental problems, and that causes ataxia problems. So we'll stop there.